Hey, thanks, Jake. So how do you change slides?
The green button on the thing you're holding.
Oh, cool. Yes. Hi. Hi. So yes, I'm David Basanta, and I've known Jake for a while. And I'd like to start obviously with the usual thing of thanking the organizers for this meeting. I know that we all got the invitations only a few weeks ago, but I know that his meeting has been planned for a long time, before COVID even. So grateful to them. I'm also grateful that I am the first contributor speaker today, because it means that I'll present you some stuff, and we have 2.5 days. So you guys hate it, you can throw things at me, and you love it, you can come and talk to me about this. So yes, I'm also a bit unhappy about the fact that I'm the first person that comes after Jake. He's one of the most, if not the most charismatic speakers I know. So it's a bit of a challenge to be the second person coming after. So I'll just throw a few things out there just to make my talk look more important than it is. Yes, so I'm the first contributed speaker. So yes, that's something. I'm the first David talking today. You're welcome. Also, I'm probably the first presenter whose slide is a lie. But I'm not going to let this derail my presentation. So let's get going. Yes, so I like to obviously say the obvious thing as well, which is that the work you see today is not something I have done myself, it's something that other people have done. So these are the people in the group. It's not necessarily a very recent slide, but all the faces here have contributed to some of the things I'll be saying today. And I want to say that not only they've done the work, they have contributed with ideas. It's for me very important to highlight the fact that I basically see my job as doing 4 things. I like to associate myself with smart people. And if you do that, then the next thing is you don't tell them what to do. You just let them do things and you try to help them. So I try to get funding that would allow them to do the work they want to do. I find great experimental collaborators that can work with. And finally, I create an intellectual environment where they can actually develop ideas. And my ideas are just basically sort of like a foundation where they basically go and do something new and different. And the ways I think about my research are cancer evolution is important. It's something that I'm not going to be, I already know the first person talking about this. I'm not going to be the last. It's going to be one of those things that will come through this presentation and many other presentations in this meeting. So it's obviously cancer evolution is something people have been thinking for a long time. Clinically relevant only for the last maybe 12 years. People have been thinking about how to use this clinically, so that's great. And there is a lot of emphasis on trying to look at the genetic aspects of cancer evolution, trying to figure out what changes in terms of mutations and pathways as the tumor evolves and changes over time, and maybe as it reacts to treatment. So I feel like I'm contributing something slightly different by saying like, well, what about the environment and the ecology of it as well. So there's many ways to look at ecology, and I like to be very literal about the whole thing. So I try to model this ecology in a more explicit way. So you were basically a cancer biologist and you're trying to understand something as basic as why is the first kind of moth, the white one, super prevalent at some stage and then gone away? And why is the second one, the black one, super scarce at the beginning and finally super prevalent? What's going on? If you were a typical cancer biologist studying this problem, and you were like very traditional, you just look at the genes and you'll find out that there's actually not a huge difference between the white and the black one, potentially just one gene. And if you were slightly more advanced in the cancer evolution pathway and think, okay, you know, there's going to be something of a phenotype, you look at the phenotype as well, you know, obviously one of them has a different color, you put them together, it's like, well, they fly about the same speed, they eat that much food, they proliferate just as fast, they have similar lifespans. So what is the difference between these two? And the difference is the industrial revolution, and clearly the environment changing in such a way that makes having the white phenotype here a severe hindrance. You are very visible to predators, right? So trying to model this context in a more explicit way is a very important part of the work we try to do. So going back to this, I was going to introduce the game theoretical work that's been done by Grace Wolf, which is a USF undergrad student who's been working with us for the last year and change. And it's something I was very excited to talk about, and especially with Jake, because there is a game here. There's a game where we talk about the interactions between fibroblast and tumor cells, which very simplistically we assume to be either resistant or sensitive. Then again, a very strong simplification, but we thought there's value to it. So we have a payoff table which we designed with the help of Andrew Marusek, and we spent a lot of time analyzing and playing a lot of therapies with it. Great, lovely work. We decided that it was time to start challenging this model with biological data. So we talked with Andrea as well. And this is something we started doing recently, and the data has destroyed the model. So my idea of I'm sure that has never happened to any of you on the history of mathematical modeling. So that's another new for us. Anyway, so the thing is, like, the idea of my presentation, which was sketched a few weeks ago when I was just thinking, you know, this is going to be great, and this is something I'm going to be introducing as a new project for this meeting, changed. So did my title. But I still have lots of ideas, and if any of you is interested in game theory, I know Jake is one of them, but I know that other people are as well. I'm more than happy to spend the next couple of days talking about the sort of ideas we had, because it's not just about frequency dependent, but density dependence as well, and the interaction with them too. So pivoting to a different project, and I decided to go to something safer, which is the work of Anna Miller, which is a former postdoc in the group who is now working at Certara as a senior scientist, who has spent many years coming with an agent-based model. And I'm just going to do an assumption here, which is that most of you, all of you hopefully know agent-based models, because this is a very sophisticated mathematical model that has been developed for many years, including the help of many experimental biologists of the group of Connor Lynch and Moffitt. And I'm going to skip all the details, because I thought that if, given the time I had, and then I had to prepare this presentation, either do a really bad job of really rushing through the details, or just going to assume that you guys know what an ABM is, that you know how complicated this ABM could potentially be just looking at it. And if you have any technical questions, you can come to me after this talk, and I'll be more than happy to walk you through every detail you might have in mind. So this is a mathematical model of multiple myeloma, which is a bone that grows into the bone, for which there's treatment, but there's no cure. And we do spend a lot of time trying to capture the ecology of the bone. So this is just basically this agent-based model where you can see every pixel is a cell type. The white stuff in the middle, that is trabecular bone. You see lots of red cells and blue cells. What they do is these are cells from the bone ecology doing the regular thing, which is destroy bone and resorb bone and produce bone, which is what healthy bone does all the time. This is a normal part of the process, and it's something that cancers will later on going to take advantage of in order to grow faster, right? So there's a lot of stuff going on here, and this model, compared to the one that Jake presented before, is super complicated. And the reason we could get away with this is because we really have spent a lot of time not only debating every single rule with biologists, but also capturing biological data would allow us to motivate, calibrate, parameterize. Now, back to some of the questions we had before. I don't know who the person who asked those questions to Jake was, but it's very important to me to realize that because this model is a lot more complex and more involved in terms of ecology, doesn't make it a better model. It's a better model at answering different questions. So this model is very detailed, and it gives you this very nice window into what happens in the bone, but even with all this bigger complexity, is still far from the amount of complexity that real bone and real bone cancers have. So this is an absolute simplification of the really complex reality that that is going on here. So once we created this model, and once we calculated the model so that it actually recapitulates bone homeostasis and what normal bone cells do, we can go and say, okay, so what happens when there are myeloma cells? And this is just one example of the many stochastic models. So simulations look a bit different from one to the other, but you can see how the tumor starts growing and takes advantage of the fact that there is a lot of bone remodeling, which means lots of factors like rank ligand and bone-derived factors that help the tumor grow faster. Like you can see a lot more density the closer you get to where bone remodeling is taking place. So the interesting thing is that we traditionally look at these things as the vicious cycle, so that there is a lot of bone remodeling going on. Factors are exchanged as a result of that. factors help the tumor grows faster, the tumor interacts with these bone cells even more, so it creates even more bone remodeling, so basically even more factors, so even more tumor. That is why it's called a vicious cycle. But these factors are something that do a lot more than just providing with extra growth for the tumor cells. They also help with treatment. And one of the things we have modeled in this approach is bortezomib, which is the standard treatment for multiple myeloma. There's a lot of biology that we use in order to motivate not only the way that bortezomib interacts and sort of hinders tumor growth, but also interacts with normal cells in the bone ecosystem. But one of the things we did is that proliferation and survival near bone, for instance, under no treatment is facilitated by these factors, but also they help the multiple myeloma cells survive the presence of bortezomib. So we try to model that. And you can see a simulation. Oh, how do I let me see the mouse? So you can see a simulation where the environment, and this is one of the things you can do with map models, obviously you can switch protection off. So the environment doesn't produce protection. So we basically now make the assumption the environment is not helping at all with the tumor when treatment is applied. Whereas on the right, protection, that is basically what the data is telling us, that there are elements in terms of the bone-derived factors and in terms of the interaction with cells in the bone environment that help the tumor maintain presence even in the presence of treatment. So what you can see obviously is very clear. If there's no tumor resistance, if there's no intrinsic resistance, simulations on the left, they just go away. Simulations on the right, they maintain this minimal residual disease, which is the fact that as long as these tumor cells are protected by the environment, they can keep proliferating and dying and proliferating and dying, just because the environment is providing these small islands of protection. The moment that cells step out of these islands of protection, that protection goes away and tumor cells die. So because they're still sensitive. That's all well, but obviously we know that there's a lot of going on in terms of resistance to treatment that is not just environment. The standard one is tumor cells that are intrinsically resistant to treatment. Now, how these tumor cells emerge is something that Jake talked about before. If it's very costly, it's hard to imagine how would it actually appear in a population when there is no selection for resistance because treatment hasn't been applied yet. But we assume that there is mutations that allow for these tumor cells to acquire resistance to treatment. And that resistance is not free. So on the left, there's no treatment, so tumor cells just do the thing. In the middle, you have environmental mediated drug resistance, which basically means that the environment provides protection in certain parts of the environment that is in contact with certain bone cells and in contact with bone-derived factors. So the more bone-derived factors, the more protection. And then you can see the green cells, which are sensitive myeloma cells, and you see black cells, which are resistant myeloma cells. And if you are a clinician and you look at the center simulations and the right simulations, you'll see very quickly that there is not much of a difference to it. So that would motivate the question, why bother trying to understand environmental mediated drug resistance if you run lots of simulations with the EMDR and lots of simulations with a no EMDR, the results are not super different. Well, obviously there are differences. The more EMDR you have, the more likely that your simulations will lead to resistance. Because then again, because resistance is costly, if there's no selection for resistant phenotypes, in the absence of treatment will basically go away in many simulations. But in many other simulations, they'll still stay there by the time you apply treatment. And because you select for them, and because they can grow where sensitive cells cannot grow, sensitive cells can only grow in the areas that are provided protection for, the difference is very minor. But the difference is actually not minor when you start looking at how this works. So these are Muller plots, and Muller plots allow us to figure out the evolution of a particular evolutionary population. And you can see on the left, you start with one single tumor cell, and you see on the right, there's a lot of cells. So you look at the no treatment, for instance, we assume two different mutation rates, and you can see that the tumor just grows. There's no treatment. There is no resistance to speak with because there's no reason for resistance to emerge. Resistance is costly, so the populations that become resistant are less efficient than non-resistant populations. The tumor is just blue, which basically means the initial population, which is just sensitive to treatment. Now, if you look at the simulations on the right with no EMDR, what you can see is the opposite. There is treatment here, and you can tell immediately whether it's treatment because there's this huge bottleneck. And after this huge bottleneck, the only cells that are resistant are the cells would basically will come up and repopulate the tumor. So you see this huge bottleneck. There's a small number of cells. If there is any resistance, there's a small number of cells. So there's this funnel effect by which whoever happened to have a resistant mutation is the funding of the emerging resistant tumor cells. And whatever positive drivers, negative passengers will have, that is what they have. That's what they start with. There is nothing else. If you look at the EMDR scenario, though, the scenario is quite different. First, the bottleneck is much wider. So the tumor doesn't really get wiped out, or at least it doesn't get that close to being completely eradicated. But the most important thing is because we have provided all these islands of protection, the heterogeneity of the initial tumor is conserved to a large extent. Not only that, these islands, because they are still allowed for the tumor cells to proliferate, that means that the cells can proliferate, potentially explore new mutations, try to explore the environmental side of these islands of protection, mostly die. But once in a while, a lucky one will actually get the right mutation, step out of the islands of protection, and start colonizing the rest of the environment. So there is a lot of different places where evolution can basically safely play with different mutations to figure out which one's actually more likely to provide protection. So not only there's more heterogeneity in the tumors where there is environmental mediated protection, there is also likely less cost of resistance as well. So these are all important factors to us. Now, I don't have a lot of time, so I'll just move on and say a few things, if this will let me. Yes, this is in bioRxiv. I'm happy to talk in the next few days, but please go ahead and take a look at the paper. It's now under review at a journal whose name starts with Nature, and unfortunately does not stop with Nature. I'm not the only one from this group making a presentation. I actually have two members of the group here, Gosia Wee and Tatiana Miti. I was told that Gosia will be the last person presenting on Wednesday, but she's actually presenting today. So make sure that you go on and see her presentation. And Tatiana is presenting on Wednesday. And with all that, I have a special issue. Please do talk to me if you have something about cancer ecology and evolution that you want to publish. There's a deadline on October the 9th. And we have a PhD program in Moffitt. So if you have PhD students who want to do mathematical oncology, we are one of the better places to do that. Thank you.
Time for 1 or 2 questions.
Another really interesting talk.
Thank you.
So, I fully appreciate that slowly cycling cells are resistant, and I believe I'm understanding correctly that that's the cost that people have been. But there are also resistance mechanisms like MGMT promoter methylation that are not associated with a decrease in proliferation rates. In fact, so there's no cost in terms of growth for that resistance mechanism. I'm just wondering how those kinds of resistance mechanisms fit into the modeling, like folate cycle variations that make cells resistant to methotrexate or aminopterin, or MGMT makes them resistant to alkylators. So I'm just wondering how that fits in when you don't lose fitness through a resistance mechanism, you just gain fitness.
Yeah. No, those are very good questions. And we just made an assumption that this was the kind of resistance would emerge. We know that in many cases, there's a cost of resistance, but you're completely right, not in all cases. Now, the point these results I think are trying to make is that even if it's costly at the beginning, because we have these islands of environmental mediated drug resistance, you have these islands where tumor cells that are sensitive. By definition, there's no cost to that. They keep proliferating, acquiring different mechanisms of resistance. So we don't assume that there's only one mechanism of resistance, and we basically make the results seem to suggest that this mediated resistance would allow for different mechanisms of resistance to emerge, because there's many places where it can emerge from. Whereas in a tumor that has not been exposed to treatment before, you really would need a really strong sort of core presence of these tumor cells that are resistant to sort of because basically the treatment will eradicate everything else, right? So you have this incredible bottleneck by which everything left is just either completely resistant or gone. So yes, I agree with you. The way we're thinking about this is that EMDR would definitely find the most effective, the least costly mechanism of resistance.